In this overview, we will cover the different compartments of the cell, protein movement through the biosynthetic pathway or the secretory pathway, movements of cargo between these different compartments via vesicle budding, and how these vesicles are targeted to the different compartments. And lastly, we will show and talk about the structures and the nomenclature of the different intracellular compartments. Inside the cell, there are several different types of compartments. For this lecture, we will discuss the intracellular compartments involved in the secretory pathway, where secreted, lysosomal, and membrane proteins are synthesized. The secretory pathway initially starts with protein synthesis in the cytosol. During protein synthesis, the entire ribosome is specifically targeted to the endoplasmic reticulum, or ER. The initial steps of asparagine-linked or N-linked glycosylation occur in the ER, as well as protein quality control to ensure that the newly synthesized proteins are properly folded. Upon leaving the ER, the proteins traffic to the Golgi apparatus for additional post-translational modifications. From the Golgi, proteins in the secretory pathway essentially have two options. They can either be targeted to the lysosome, or they can go to the plasma membrane. All of the compartments of the secretory pathway are made up of phospholipids, or at least the compartmental boundaries. Phospholipids are basically a polar head group attached to two hydrophobic tails. And phospholipids create these compartments actually quite spontaneously, as the hydrophobic tails are bearing against each other, avoiding water, and then protruding out the polar head group facing the water. Shown on the right is a schematic from molecular biology of cell of how initially a planar phospholipid bilayer is formed, but however now the edges are exposed to water, and so in order to prevent those hydrophobic tails to interact with the water, the planar phospholipid bilayer curves and eventually makes a sealed compartment made up entirely of phospholipids. The water-filled insides of intracellular compartments of the cell are called the lumen. Thus, if you take the Golgi, for example, the inside of the Golgi is called the lumen, and of course the material or the water facing on the outside is actually the cytosol. Water-soluble proteins in the cell are moved from compartment to compartment via vesicles. Vesicles are formed by budding, where a part of the membrane blebs off and then eventually pinches off to form a completed vesicle, shown in the middle on the left panel there. That vesicle now can traffic or flow to another compartment where it can interact with and eventually fuse, spewing out the cargo into the second compartment. In addition to water-soluble cargo, shown in, like I said, the red dots, and membrane proteins that are part of the vesicle are also transferred from compartment to compartment. And this is the major pathway for membrane proteins and water-soluble proteins to go from the different compartments, as well as to the plasma membrane for secretion or incorporation into the plasma membrane. Vesicle transport, typically in the biosynthetic pathway, the forward direction is called anterograde transport, shown on the right there, going from the ER to the cis-Golgi. In contrast, reverse transport, or going backwards, is called retrograde transport. The COP and all the details in this cartoon are not that important, those types of proteins, but this just shows the schematic of how things going from forward as well as they can go backwards in a cell. Lipid movement between compartments can go by two mechanisms. The first is by vesicle budding. Essentially, when a vesicle buds off, it takes some of the lipid components with it, shown on the left there. And when a vesicle then fuses with another compartment, it delivers not only the intracellular cargo, as well as the membrane proteins, but also the lipids that were in that vesicle. On the right shows a more specified or specialized mechanism for delivering phospholipids, where an actual protein will then take lipids from one compartment and actually transfer them to another compartment. And these, again, are to specifically distribute different types of phospholipids to the different compartments of the cell. With all of this trafficking of the different vesicles all around the cell, it's no wonder that most of the trafficking is actually targeted by some mechanism. A lot of the targeting is done by signal sequences, or small stretches of peptide that are attached to the proteins that have to be targeted to the different compartments. This table shows a series of different types of signal sequences. For example, import into the ER, shown there is a mostly hydrophobic sequence followed by a bunch of charges on the C-terminus. return to the ER, or again retrograde transport, has been shown there are two motifs. Again, the list goes on. If you notice that a lot of these have actually a very similar, either positively charged lysines and arginines, as well as hydrophobic species in the case of the import into the ER, as well as the export from the nucleus. It's not really important to memorize these, but the important concept here is that most of the targeting of the proteins is actually encoded in the protein sequence and not the vesicle itself. The next series of slides, we will actually go through the different compartments of the secretory or the biosynthetic pathway. We will see pictures of what these compartments look like from actual EM pictures, as well as cartoon drawings that you will see typically in textbooks. I will also touch on some basic biochemical conversions that go on in the different compartments. In the lecture, we will go into more mechanistic detail of what each of the compartments do to the different proteins and how the different proteins are targeted eventually to the lysosome or the cell surface. So our first stop on the secretory pathway is the endoplasmic reticulum. And the endoplasmic reticulum is a compartment, and it's actually a compartment of an array of cisternae, or flattened vesicles, and tubules that are attached essentially to the nuclear envelope, shown here in this classic textbook cartoon. The ER comes in two flavors. There is the rough ER, and it's called so, as you'll see in a moment, because of the ribosomes that are attached to it, and makes the ER look very rough in the pictures in the EM, as well as the smooth ER. So the smooth ER, actually a lot of small molecule and lipid synthesis takes place. So for example, gluconeogenesis occurs in the lumen of the smooth ER because glucose 6-phosphatase is located there. Lipid synthesis, again, lots of hydrophobic compounds are synthesized that the body needs: phospholipids, triglycerides, and steroids. In addition, not all of cholesterol synthesis takes place in the smooth ER, but one of the key enzymes, HMG co-reductase, is associated with the smooth ER. In addition to synthesis, the smooth ER is also a place to detoxify small molecules or hydrophobic compounds, and this is because cytochrome P450s are located there, or oxidases, as well as it's a place for detoxification of ethanol through microsomal ethanol oxidizing system. It's a wonder that in the liver, of course, then we have most of these cells have a lot of smooth ER. In addition, it is thought that the calcium sequestration that the ER does, a lot of that is done through the smooth ER. Here on the right, you actually see an EM picture of the smooth ER. Things that are not smooth ER are highlighted, so the nucleus shown in red and a couple of mitochondria shown in blue, but all of those circles there below those are actually smooth ER, and all those tubules. Again, you're seeing a slice here, so again, this is like one slice, and so you're seeing the different tubes of the smooth ER. Here's a blow-up again. Here's a mitochondria on the right, a vesicle on the left, and all of these tubular nature is all smooth ER. One thing that you have to remember about smooth ER, although it's important, it has no role in protein synthesis for the most part. Smooth ER is again more of a detoxification, sequestering, as well as lipid synthesis compartment. In contrast to the smooth ER is the rough ER, which is the site for protein synthesis for membrane, lysosomal, and secreted proteins. The roughness of the rough ER comes from those ribosomes which are making the proteins which are docked on to the ER membrane. If you look at this EM picture here, you can see very nicely these stacks or cisternae of ER, and the black dots are kind of giving it that rough texture. Here I will highlight one of those cisternae in blue, and again, that's just one of the many ERs that you are seeing here. In the upper right-hand corner for your sort of orientation, that's the nucleus. If we blow up this cisternae of ER membranes, you can see really nicely these little black dots which actually correspond to individual ribosomes, and some of them are docked onto the ER membrane and some of them are very close. After protein synthesis occurs in the ER, the next step on the biosynthetic pathway is to the Golgi. The Golgi apparatus are individual stacks of membrane compartments. So the individual cisternae are actually not connected as they are in the ER. The cis Golgi are the compartments that are very close to the ER, and these tend to be convex, whereas the trans Golgi, which are farther away from the ER and closer to the plasma membrane, tend to be concave. The major function of the Golgi apparatus is to perform post-translational glycosylation. Both asparagine as well as serine and threonine linked glycosylation occurs in the Golgi. One of the advantages of having these individual compartments is that you can then position the cell positions enzymes in the different compartments to do certain jobs. So different enzymes are in the cis, the medial or the middle, and the trans Golgi. The Golgi apparatus is always capitalized, shown by the asterisk, because it's named after the discoverer, as well as Nobel laureate Camille Golgi. This shows an EM picture of a couple of cells with Golgi complexes. I think you can see the stacks, if you will, of Golgi in the different types of cells. If you blow this picture up, you can actually see now very clearly the nuclear envelope here is on the top. You can see the rough ER, and again studded with those ribosomes. There are transitional elements, which means elements of the ER now converting into Golgi, and then now you see the Golgi stacks. And I think very nicely in this EM picture, you can see the concave surface here facing the plasma membrane, where the convex surface here on the outside is, or on the top, excuse me, is facing the ER. After the Golgi, membrane and water-soluble proteins have two options. They can go to the lysosome or to the surface or the plasma membrane, where if they're membrane proteins, they become resident, and if they're water-soluble proteins, they are secreted into the extracellular matrix. Our last stop on the secretory pathway is, of course, the plasma membrane. All this time, the membrane proteins have actually been inside out in all of these intracellular components. What I mean by that is, if you look at the little black dot in these compartments, those are facing the lumen side. However, when you get to the plasma membrane, the black dot is facing out. This allows those luminal enzymes in the Golgi and the ER to modify the extracellular face of the membrane proteins. And then when the vesicles butt off the Golgi headed for the plasma membrane, these lipid vesicles then fuse with the plasma membrane, spewing out their intracellular cargo, shown here as pink and dots and blue sticks. But the membrane proteins and the lipids that made up the vesicle now become part of the plasma membrane. One way to keep this all straight is to remember that no matter what happens to a membrane protein, the cytosolic side of the protein always faces cytosol, whether it's in an intracellular compartment or whether it's at the plasma membrane.